Good afternoon, and thank all of you for joining today, for joining the American Society for Microbiology for an update on the Center for Disease Control and Prevention's Advanced Molecular Detection Program, its role in genomic surveillance and sequencing of SARS-CoV-2, and additional plans beyond the pandemic. My name is Susan Sharp. I'm a past president of ASM, and I will be your moderator today. I also wish to express ASM's thanks to Senator Amy Klobuchar's office for providing logistical support for this event. Joining me this afternoon is Dr. Greg Armstrong, the director of the CDC's Advanced Molecular Detection Program, the AMD program. And it's through Dr. Armstrong's leadership that ASM has brought next generation genomic sequencing, bioinformatics, and related technology to public health in the United States. Over this past year, in addition to his role as the AMD director, Dr. Armstrong has also served as the deputy incident manager and the chief science officer for the agency's COVID response. His expertise, ingenuity, and vision have been invaluable in this effort. ASM is a proud supporter, along with many other organizations and institutions, of CDC's efforts to bring pathogen genomics to the front lines of public health. This technology enables us to detect diseases more quickly and more accurately, and allows us to identify outbreaks sooner to protect people from current, emerging, and evolving infectious diseases threats. The CDC AMD program has played an instrumental role during the COVID-19 pandemic, from tracking the spread of the virus to now ensuring we understand viral variants, including the Delta variant, and ensuring that we can stay one step ahead of them. We are especially pleased to bring you this update today because the program is in a transformative moment. As most of you know, the American Rescue Plan passed earlier this year included $1.7 billion in supplemental funding for the AMD program. We thank Congress for including these critical funds, and we especially appreciate the leadership of Senator Tammy Baldwin, Congressman Ami Bera, and Congressman Scott Peters in this effort. We also wish to thank the House Appropriations Committee on Labor, Health and Human Services, and Education, led by Chair Rosa DeLauro and Congressman Tom Cole, for proposing increased base funding for the program for the next fiscal year, FY 2022. It's going to be critical over the long term that funding for the program's base increase so that we can sustain the improved infrastructure and increased capacity that is being made possible through the emergency funding. Among other advances, the recent funding infusion has allowed the AMD program to move quickly to increase viral sequencing. The Biden administration and CDC have also outlined plans for future activities, which we're going to talk about more now. So let's get started. First, I'd like to welcome all of you and thank Dr. Armstrong for joining us today. It's good to see you, Greg. Thanks for being here with us.
Hey, no, thanks for the opportunity to discuss this with everybody. And I just wanted to take a moment to thank ASM as well. ASM has been really visionary in this area, and I want to thank them for the support. I want to thank Congress for the support that they've given us as well.
Great, thank you. But before we start talking about the program's current activities, could you take us through and provide a quick overview of where the AMD program was prior to the emergence of SARS-CoV-2?
Yeah, so just very quickly, the program that I run, the Advanced Molecular Detection Program, was established by Congress in 2014, fiscal year 2014, with $30 million per year. I'm simplifying things a little bit here, but the main objective is to bring next generation sequencing and bioinformatics into the U.S. public health system. And so we had been, this was actually at the time was quite a big job, but we've been able to, before the pandemic, we were able to accomplish quite a bit. Here at the agency at CDC, next generation sequencing and bioinformatics essentially it was already in use in all of the infectious disease programs. You can name I think, any infectious disease of public health importance. And we've been using next generation sequencing now to either characterize the pathogen or use it in some other way to inform public health decisions about it. In addition, we've been working with a couple of other, actually with a few programs at CDC to get the technology out to state and local health departments, such that by the end of 2019, all state health departments had next generation sequencing capacity, and an increasing number of local health departments did as well. We'd also set up a network of state-based regional bioinformaticians. The bioinformatics is a relatively new specialty in science, and there's a pretty stiff competition out there for bioinformaticians. But we found that we can actually attract them to public health for the same things that attract a lot of people to public health, which is the opportunity to have a big impact. But increasingly, states are also hiring bioinformaticians, and so there's really a growing network of them in state health departments. And we'd also done quite a bit of training with microbiologists out there. There was a huge amount of interest when we first started this, and there still is in the microbiology community in state and local public health labs in learning more about genomics and about how sequencing can be applied in public health. However, the work that we'd done, it was in some ways an unfinished project. So sequencing was used inconsistently outside of foodborne disease. So we'd gotten to a point where all of the state health departments were sequencing foodborne pathogens, and an increasing number were starting to sequence other pathogens, but it wasn't anywhere near as robustly used as we felt was optimal. But at the time that the pandemic started. In addition, our training for the epidemiologists had really only just begun. This has been on our long-term plan, and it's really key for us that we get the epidemiologists trained up. They're the ones that have to make use of the data. They're the ones that have to understand where sequencing is useful, where it's not useful, how it can be useful, how to interact with the lab on this. And so that was very much a work in progress. And in addition, there was real limited national infrastructure, building a structure for to allow the exchange of data and to allow the kind of analysis that needs to get done for bioinformatics. So for those of you who don't know much about the field, bioinformatics is extremely complex. The types of analysis really go far beyond what we've ever had to do in public health previously. And for that reason, at CDC, we had to develop initially a scientific computing center with high performance computing that we were in the process of starting to move to the cloud when the pandemic hit. But in state and local health departments, they have the same challenges. They've got to figure out how to first of all how to accept and how to store huge amounts of data, but also how to get access to the sorts of computing resources that they need to do this analysis. And that's where we were essentially when the pandemic hit.
Yeah, there's a lot more to discuss about training, and I'll get to that question here in the next couple of questions. But before then, we do understand from the CDC director, Dr. Rochelle Walensky, that thanks to the AMD program, that the United States has dramatically increased the percentage of COVID-19 samples that have been sequenced compared to where we were at the end of 2020, for example. So can you comment for us on how these significant changes were accomplished and how these efforts are currently ongoing?
Yeah. Again, let me just go to that slide deck. Actually, let me just pull up a couple of slides here. There we go. So just before I get into that question, one thing I always emphasize is, it's important whenever you're talking about sequencing to be very clear what the objectives are. There's one of the most common mistakes we see when in groups that we talk with about sequencing for public health purposes is failing to be clear about what those objectives are. And for us in the U.S., it's different at the national and state level. There's actually quite a bit of overlap here, but just to simplify things, at the national level, it's everything that we've been talking about in the last few months about variants. We need to be able to detect and track variants that have implications for vaccines, therapeutics, or diagnostics, or that have important epidemiologic characteristics such as increased transmissibility or increased severity. At the state and local level, like I said, there's overlap, but in addition to those national objectives, sequencing can provide a more granular understanding of that epidemiology at the local level, which can be very useful in directing public health efforts. It can also identify clusters that wouldn't be visible otherwise, and where there's potentially an intervention to stop transmission. There's no doubt that it's useful in investigating outbreaks and understanding which cases are part of outbreaks and understanding what's driving the outbreaks and so forth. And it may also have utilities for some other public health operations like contact tracing. It's a little less clear with those, but I think it might be useful. And so and you'll notice to meet these objectives, we actually don't need to sequence everything. This is a common misconception. If we're if we were going for containment, for example, then it would be imperative that we get as many samples as we can and for sequencing. But that's not really the case here, and containment, quite frankly, would be difficult with SARS-CoV-2 because there's so many asymptomatic infections or mild infections. But at any rate, so there's no doubt that the more sequencing you do, the better off you are. The more information you have. But there is a point at which you reach diminishing returns and where your resources are better served in other areas. So let me just see if I can. Yeah, there we go. So so where are we with sequencing in the U.S.? So a couple of things. First off, very early on in the pandemic, our group, there's somebody from the AMD program, Duncan McCannell, got together a national consortium of groups doing sequencing. These are groups from all over the spectrum, from public health, from academia, industry, NGOs. In fact, there are, I think, over 1,000 participants now, and I think over 200 different organizations. They got started back in April of 2020, and this group is very useful in driving consensus around standards for sequencing, on the need to make sequence data publicly available as soon as possible. They were able to get consensus on that very early on. And it's also been extremely useful in sharing information about the pandemic and lessons learned from various groups, so they can accelerate the innovative process that's needed to optimize the sequencing and the bioinformatics to support it. So who's doing the sequencing? So first off, state and local public health labs, they've been sequencing up to about 8,000 per week. All these numbers were sort of at the peak. They've come down in the last several weeks because cases came down so much. They're starting to come up again, unfortunately, because cases are coming up again. At our peak, they were sequencing probably about 8,000 per week. Back in starting in December, because when it became clear that variants were a serious risk with the emergence of B117 in the UK, we funded 9 different reference labs, and they've been sequencing up to 25,000 per week. Academic labs and industry is also sequencing up to about another 8,000. There's also 29 different groups that we've funded through CDC through what we call broad agency announcements. And these have actually been quite useful. I can talk a little bit more about those later if there's interest. But these are groups. Oftentimes they're working with state health departments. In fact, most of these are working with state health departments, and a lot of them have very specific niches that they fill. Like one of these in Wisconsin was looking early on at transmission in institutes of higher education, and actually came up with some very useful data that for universities that was helpful for them in reopening. We have a group now in Mississippi, the University of Mississippi, that's actually working with the health department there. That's helping them respond to the current pandemic there. Finally, there's something we call National SARS-CoV-2 Strain Surveillance, NS3. This is a much smaller number. It had peaked at about 750 per week. It's now down to about 200 per week. But what's important about this particular thing is that we actually, through NS3, we actually get the samples themselves, so that we can grow up the virus and do some additional characterization on that. You can get a lot of information from the sequencing, but that only gets you so far. At some point, you actually have to grow up the virus and do the testing in the lab. But we don't just do it ourselves. We also take any of those viruses that are unique for whatever reason, and we make those available through something called BEI resources to industry and to academia and to the vaccine makers, for example, so that they can do the testing that they need. So this is actually quite an important national resource. In addition to that, these are all sort of for all these systems are useful in background surveillance, but it's very useful beyond that to get sequencing of specimens from specific cases where that are going to be useful in decision making down the line. So, for example, integrating we've at the agency there's a number of epidemiologic surveillance systems that look at hospitalizations, for example, and so we've gotten sequencing integrated into those because we're actually very interested in knowing the variants that are associated with more severe disease. At the same time, we're also including those in vaccine effectiveness studies and vaccine breakthrough studies, so that we can get a better sense of whether these are interfering with the emerging variants have implications for vaccines or not. Fortunately, so far they don't. But I think I have another slide somewhere on here. Yeah, this, by the way, this just shows how much we've scaled up sequencing since December, since right about here. We've divided the sequences that we're getting that we use for surveillance into 4 different categories. In the light blue on the bottom are specimens that are done at clinical and academic laboratories. Again, all these data are publicly available, so we use them, researchers use them, the vaccine companies use them, the diagnostic companies use them. The state territorial labs are here in dark blue, and you can see they've increased quite a bit since December. Private sector labs are here in pink. And then, in addition to those, these in orange are these contract labs that we've contracted with. Most of these, again, are private sector labs. Some of them are academic labs. But you can see how this has evolved over time. The numbers have come down, like I said, because it's actually most of these labs at this point are sequencing all the positive specimens they can get a hold of. One of the challenges we have in the U.S., the challenge at this point is it's actually not the sequencing. There's plenty of sequencing capacity. We actually have quite a bit of excess sequencing capacity at the agency right now. The challenges in the U.S. is everything that comes before and after sequencing. The lab market is very dispersed, and so to get specimens, you end up having to go out to a lot of different labs. This is one of the challenges that state and local health departments have as well, is getting the specimens from the lab before they discard them. The typical lab just doesn't have the resources to hold the specimens after they've tested them. But at any rate, these, as I mentioned earlier, sadly, the number of sequences is starting to come up again because the number of cases is coming up. So let me stop there.
Okay. Just a couple of follow-ups to that, if you don't mind. We've talked about the variants, and we've all seen the reports about the Delta variant and how it's now the dominant strain in the United States, particularly in the areas, as you mentioned, that have low vaccination rates. It's where we're seeing this Delta variant pop up. So I know you spoke a little bit to this, but is there anything else that you'd like to explain to us about how AMD is providing information on those variants? What can we expect? And again, why is it so important for all of us to understand how important this work is for sequencing and variant analysis?
Yeah, so within CDC, there's a discrete group that responds to the pandemic. It's the emergency response effort that I and others have rotated through. And there's a group in there that actually manages the laboratory aspects, the laboratory task force. And within them, they've got a variant surveillance task force that we work very closely with. So when things started back in December, we in the AMD program we're doing most of the sequencing work. We've at this point have transferred quite a bit of that over to the laboratory task force, which is managing the day-to-day aspects of this as of now. And they've really increased quite a bit, and I think done quite a good job of making the data available on our website, including what we call nowcast. So there's always a delay in getting the sequencing data. So with these contract labs, for example, these large commercial labs that we work with, they have a very large network of laboratories, and they typically have to get select the samples, the plasma samples. They have to get them to a centralized location. There's oftentimes multiple information systems that they have to integrate the data from, to the epidemiologic data, the data about the case that they need to send to us along with the sequence data, and so there's always a delay in getting that data, and then we use these nowcasts to project forward to what we think the current prevalence is, and just it's about to be that site's about to be updated with the latest nowcast, but it's you can when you go to the site you can just see the trends itself. It's there's probably greater than 80% of all cases in the U.S. right now are Delta, and we're anticipating that's going to increase. We've been lucky so far here in the U.S. in that the variants that we've seen and have become predominant, like B117, which is also called Alpha, or the Delta variant, they're actually susceptible to the vaccine. The vaccine is all the data we have so far is that the vaccine is very effective against those. There's still some data that we're waiting for. When a new variant emerges, the first piece of data we get is the antibody test. So they take serum from people who've been vaccinated and they see whether that neutralizes the virus or not. And they do that very quickly, and that's been done in several labs right now. And the data is very consistent that the serum from people who've been immunized with the U.S. vaccines is very effective against against Delta variant, and it was also very effective against B117 and the other variants that we've seen as well. The best data is the data from epidemiologic studies to see, among people who've been vaccinated, do they have lower rates of infection with these new variants? And those sorts of studies are complex. They just logistically very complex. They take a long time to get the data from, and so we don't have those data back yet. But so far with the other variants, what we've seen is that those track very well along with the neutralization data. So we're very optimistic that the vaccine, everything we've seen so far is that the vaccines that we're using are effective against the Delta variant. And in fact, where we're seeing Delta emerge in the greatest numbers are in those areas where vaccination rates are lowest. And as Rochelle Walensky, the CDC director, said, unfortunately, this is becoming a pandemic of the unvaccinated. Now, what we've been concerned about all along is that variants are going to emerge that might not be susceptible to the vaccine, or that might have mutations in those areas of the virus that are used by diagnostics. Mm-hmm. Or that, for the therapeutics like monoclonal antibodies, that variants will emerge that are resistant to those. And in fact, we've already seen that for some of the variants. And so I think, quite honestly, it's probably just a matter of time before we see that. But we haven't seen it yet, as far as we know. And so, but by no means should we let down our guard at this point. We actually, this, variant surveillance is here for the long term. This is going to be a long-term part of public health practice is variant surveillance, because we need an early warning if variants emerge that have implications for vaccines, diagnostics, or therapeutics.
Along that line, Greg, can you tell us how do you know where to go to take a good representative sample from the United States? We're a very large, diverse country. What are the challenges to accomplishing that? And how do you know that we're picking the right samples to sequence, since we're not sequencing, at least early on, we weren't sequencing everything, to make sure that we can pick up any additional variants that might enter the U.S. or develop inside the U.S.?
Yes. So how do we know that that the data that we're getting is really representative of the U.S.? As I mentioned, we don't need to sequence everything, but it does need to be fairly representative. I mean, it doesn't need to be perfect. We have years of experience. We have actually decades of experience with influenza surveillance, and we know to pick up important emerging influenza variants we don't need to sequence everything, which would be impossible anyway with influenza. But but we also know that the surveillance doesn't need to be perfect. It just needs to be good enough, quite honestly. But that issue of representativeness was a concern of ours from the beginning, and it is still a concern of ours. The reason we went with 9 different labs, we could have actually gone with a smaller number of contract laboratories and gotten coverage for most of the U.S. with them. But we had concerns that there were differences in the markets these laboratories were serving, and that might that might impact the representativeness. And so we opted to go for a larger number of laboratories. We also went for some of the smaller laboratories that serve parts of the country that weren't as well served by some of the larger laboratories. But we try to get data from every state, and the future is that the states are going to be doing sequencing themselves, I think, or they'll be getting the specimen and sending to us for sequencing centrally. But I think a lot of the sequencing in the long term is going to be done in states. And we've been, mostly successful at getting, adequate numbers from states. We're not quite down to the state level yet. We're at the regional level where we feel comfortable that the numbers are stable enough, representative enough. And that's the data that we show on our website is the breakdown by region. But you can see even there, when you look at the regional differences, there are regional differences. Fortunately, at this point, they don't have big implications for public health practice. One of the things about the variants we've seen so far is that the good news is that the public health interventions are the same for each of them. It's vaccination, isolation, quarantine, physical distancing, and so forth. So we think that what we're getting is representative enough, and we'd like more granularity in this. And in fact, we're looking at what we can do to expand the number of specimens that we're getting from certain areas where we aren't getting a lot. And we're also we're in the process of taking a hard look at our data right now. We don't have the results yet, but for example, between these different labs, a lot of these in probably most states, we have multiple labs serving, getting sequencing from the same state. We're in the process of comparing those to see if we're actually seeing any differences between those, which will tell us how important it is to, how likely it is that the data we're getting is representative.
Yeah, and when hearing so much more about the variants, especially the Delta variant, which we've talked about, there's been some talk about the need for potentially having booster shots for against SARS-CoV-2. So how is the information that you guys are gathering through the AMD program going to help in studying the variants and in deciding on studying vaccination as well?
Yeah, that's an excellent question. And you have to keep in mind, we have now about 1.5 years of experience with coronaviruses that, cross the species barrier and cause pandemics in humans. This is the first time. We have by far a lot more experience with influenza. So, as you all know, the influenza vaccine varies from year to year. There's actually a different vaccine formulation for the Northern Hemisphere, and then 6 months later, they make a different formulation for the Southern Hemisphere, and so on and so forth. And it's because influenza mutates so quickly. It mutates fast enough that on that sort of time scale, when every few influenza seasons, a new variant will emerge that isn't covered by the current vaccines, and so they have to change the vaccine composition. We don't know yet what to expect from coronaviruses, but this is what we do know is that they do mutate like influenza, but at a much slower rate, probably about 1/3 of the rate. Now, does that mean that we'll need a different vaccine every 3 years? We just don't know at this point. No matter what anybody tells you, we won't know until we actually get experience with this. But what we do know is, partly because of the funding, actually in large part because of the funding from the EMD program, the flu program at CDC years ago, one of the first projects we funded was for them to move to what they call a sequence first approach, in which rather than doing the traditional characterization of the influenza viruses, so they get about 10,000 samples. It varies a lot from year to year, but from around the country, and they also work with a number of countries around the world to characterize those. To characterize those in the lab. But what they've found early on when they went to the sequence first approach, where instead of like doing that very labor intensive laboratory testing, what they did was they started sequencing first, and that sequence data actually help them really pointed out samples that had viruses that were of particular interest. So viruses that part of the phylogenetic tree, for example, that might be pushing out that, a part of the phylogeny of influenza viruses that appear to be emerging that are a higher priority for characterization. And this is, I think most countries now, at least certainly most of the larger countries, have moved to the sequence first approach for influenza. And I can tell you the people there's a meeting in Geneva every 6 months to make these decisions about the Northern Hemisphere and then the Southern Hemisphere vaccine. They feel that the data they're getting is far better than what they've been able to get in the past before we had all this. I think with SARS-CoV-2, we're going to see the same sort of thing. We're going to be sequencing this virus. We're going to be looking for signatures in those sequences that suggest that this virus should be a high priority for some reason. And then we're prioritizing those viruses for further characterization or for further epidemiologic investigation of those cases, so that we could get an earlier reading on whether there's an important variant that's emerging that's going to have implications for vaccines or therapeutics or diagnostics. So I think I don't think it's going to be as fast a pace as influenza, but I think there's a good chance that we are, at some point down the line, I can't say when, going to need boosters of, for a variant that has diverged so far that the current vaccines don't cover it. But that's speculative. We just won't know till we have the experience. We won't know until we actually see that variant emerge.
Yeah, that's great. That leads into kind of my next question about the significant disparities that we've seen with COVID-19 as far as morbidity and mortality across the United States. And this is a big concern to Congress, to ASM, our members, and other scientific societies. So how are you thinking about as you continue on and ramp up with your sequencing, how are you going to try to connect that genomic data with the epidemiologic data?
Yeah, so and that's actually a key point. So we haven't seen any data yet that there are significant differences, in the variants in various populations in the U.S. that are accounting for these disparities that we're seeing. The disparities are clearly there and are clearly important and clearly need to be addressed, but they need to be addressed with vaccination and with other interventions. This has been an issue though that has been a little bit problematic for us. So the largest volume of data that we've been getting, the largest sort of most highly reliable volume of data that we've been getting have been from these contract labs over the last several months. And there is real limits to the epidemiologic data that they can get. And so, for example, race ethnicity is often just not available to them. They're all trying to get it, but it's just oftentimes not in the information systems that they have access to. And so we've actually made a real push to get in place a automated system to get the sequence data to state and local health departments, where they actually have much more robust case data. That includes race, ethnicity, but also like more granular location data and other pieces of data that are going to be important in addressing these disparities. It's a complex problem just because the informatics part of this is complex, and because we're dealing with 50 state jurisdictions plus District of Columbia plus a lot of large local jurisdictions, each of which has a different information system. And we're also working with 9 different labs, all of which have different information systems. In fact, within those labs, oftentimes they have multiple information systems. So it's actually a very complex problem, but it's something that we're actively working. At this point, we haven't seen that there's that the variants themselves are causing any of these disparities. They're unfortunately highlighting the disparities, especially right now, highlighting where vaccination coverage is low.
Yeah, well, in talking about, you mentioned a couple of times now about the contract laboratories and the other laboratories that are helping out and doing some of the sequencing. And there has been a lot of talk about data sharing and the challenges, between data sharing between public health laboratories, academic laboratories, clinical laboratories, and industry companies even. And Congress has been working hard to support the data modernization initiative at the CDC. And I'm wondering how this work intersects with the AMD sequencing works in terms of facilitating rapid data sharing, how those efforts are ongoing currently, and what challenges you see that you've already had and that you might see in the future.
Yeah. So excellent questions. Trying to figure out where to start with that. So first off, in terms of open data, from the beginning in the EMD program, where we've been pushing for data to be publicly available whenever possible. And one of the advantages we have is that microbial genomic data, in most cases, is not considered confidential. It can be made publicly available without the risk of the loss of confidentiality. The issues come in around what we call the metadata, the data about the individual case. For example, if you were to release the individual age, age 23, rather than the age group, 20 to 29, for example, for certain pathogens. And if you release data that's too fine a granular level, then that becomes a problem as well. So the issues that we've had to deal with have mostly been around the metadata and making sure that any of that metadata that goes along with the sequence data is not so granular that it could potentially somewhere down the line compromise confidentiality. And in fact, all of the groups that we fund at CDC, we require them, unless they have a waiver, to make their data publicly available, the genomic data publicly available within a certain period of time. NIH, by the way, does the same thing for researchers who they fund. So all of our foodborne disease data actually has been publicly available for years. FDA does the same thing. In fact, we work with NIH to integrate this data and get some regular analysis of the FDA and the CDC foodborne disease data together. The influenza data we make publicly available. There's the TB data is made publicly available, but after a delay for a year, it just there's that's in order to increase our ability to make it confidential. The HIV data we don't make public just because there are a lot of issues around confidentiality around that. But in general, there's been a consensus to make the data publicly available, and which has been a great boon for researchers, for industry, for public health. The managing this data, managing public health data in general, public health, has really fallen behind in this area, or had fallen behind in this area. Especially on the epidemiology side, which was why, for many of us, this data modernization initiative and the funding again that came from Congress to make this possible has been so welcome, because there is throughout the public health system a real need to modernize our data systems. And this is coming at a time when the technologies for doing this have advanced, to a point where there's also just a lot more opportunity than there was like 10 or 20 years ago. for this. There is a separate set of funding that came to CDC for data modernization, which is focused on this epidemiologic data. We've been working very closely with them from the beginning. In fact, we met with them for 3 hours yesterday this is a series of meetings that we have around how we're going to make sure that we stay together on this. For this genomic data, for it to be most useful, in order for it to be useful at all, we really have to be able to integrate it effectively with the epidemiologic data. And that's a challenge. It's a challenge because the information systems are oftentimes very different. It's a challenge because the epidemiologic data is just so diverse. If you think about the type of data you need, to investigate a foodborne disease outbreak versus the type of data that you would need for, tracking TB cases. It's just completely different types of data. They're oftentimes structured very differently, which is again a need for the data modernization initiative. In addition to those, to these challenges, the tools that there are for integrating this data, for bringing this genomic and epidemiologic data together, are still somewhat rudimentary. They've been advancing actually during the pandemic. There's been a real need for those, and so there's some groups that we've been collaborating with for years, like the NextString group at the University of Washington. Or there's a group at Oxford University that's also been actually developing some very useful tools. But we're hoping in the future, part of what we're planning on doing is in the long run, with this funding is to develop a more robust collaboration with academia, including figuring out where these tools are being developed that could potentially be useful in the long run for public health. Figure out how we can use this funding to accelerate the development of those.
And Greg, if I could ask you to put up your one of your slides again, I think it was your slide number 5. It talked about the SPHERES program. So the Advanced Molecular Detection Program built this unique public-private consortium called SPHERES. And SPHERES was developed to leverage and build on existing AMD infrastructure and expertise to bolster our pandemic response. And this partnership continues to draw, as if you've talked about, from all sectors, bringing together public health as well as clinical laboratories, academic research institutions, and private sector entities to work together to advance viral genomic surveillance. So how do you envision a consortium like this working even beyond COVID-19? And what do you think with partnerships like this that you'll be able to do in the future that you've not been able to do up until now?
Yeah. From the beginning of the pandemic, actually from the beginning of the AMD program, we've really pushed collaboration with the academic sector, with universities. This is partly a necessity because at the beginning of the AMD program, we just didn't have anywhere near enough experience with sequencing and bioinformatics, but also we just viewed it as a real opportunity. With the emergence of next generation sequencing and pathogen genomics, there's been a real increase in interest in universities in public health, which is great. I mean, I've been in public health for 20 plus years, actually going on 25 years. And I've never seen this level of interest in this, and we wanted to take advantage of that. One of the challenges is that the academic groups get funding mostly for clinical work rather than public health work, and that's mostly because NIH has always had a very large budget for extramural research, and CDC has never had sort of that large a budget to support public health research. And so, the academic groups would get interested and do work on, pathogens during public health emergencies, and might even do some collaboration with public health departments. But that would fall off in between because they didn't have any support for that. We found that really from the beginning of the EMD program, supporting those interactions has really been helpful. The state health departments, a lot of these collaborations have been between local universities and state health departments. The health departments get access to, newer technology, and the academic groups get access to, oftentimes they need access to samples, when it comes to sequencing and the data that goes along with those, and so they can work out those, the data use agreements and material transfer agreements and everything they need to work together. But it also gets those academic groups more interested in public health, and the students that are coming up through those more interested in public health, which is very useful to us in the long term. When the pandemic hit, that interest skyrocketed, and part of the reason that Dr. McCannell from our group developed the spheres was to take advantage of that, and we really want to build on that in the long term. The spheres right now is essentially a weekly call. There's more to it than that, but there's a call every Wednesday afternoon for an hour, and there's at this point I think there's probably 200 or 300, sometimes up to 500 people attending it, and there's just so much interest in this, so much interest in exchanging information. And we found it's hugely useful to have these different sectors on the phone, the public health groups, which help sort of, they're oftentimes just talking about their own work, but they also help state what the priorities are and what trends that they're seeing. The academic groups, which are talking about what they're doing. Industry is actually also doing quite a bit of sequencing, sometimes with funding from us, sometimes for other reasons. And the NGOs, there's several that join us as well. Like the Gates Foundation we've been working with them for a number of years. They've had a very big interest in promoting the use of microbial genomics globally, and so they and the Rockefeller Foundation, there's a few others, are actually learning from these experiences that we're getting from public health in the U.S. as well as what we're learning from these other sectors, and so just that exchange of information has been, I think, extremely useful. We haven't figured out how we translate this into a long-term program. For right now, it's not slowing down at all, and unfortunately, it's increasing because of the increase in cases. So I don't have a pat answer for it, but I can tell you we're very interested in figuring out how we can take the lessons learned from spheres and continue that kind of collaboration on some sort of level in the long term.
Yeah, I know we've got about 5 minutes left, and I got two really important questions that I need some brief comments from you about. So, and the first one is about the American Rescue Plan money will also allow the CDC to establish 6 academic centers of excellence for genomic epidemiology. So can you just briefly, because I have another important question too to get to, tell us a little bit more about those centers, what challenges you anticipate, what else Congress can do potentially to help this be a success, and then lastly, the timing for when these centers will be stood up.
Yeah, so they're going to be competed out, 6 centers, and at each of those centers, it's going to be a combination of a public health agency, probably a state public health agency, but there in some cases it might be local public health agencies, or consortia of public health agencies working with one or more academic labs. And these centers of excellence sort of bring together everything that I was talking about before. Their main task is going to be to help us bring genomic technologies and other related technologies into public health, and to foster the innovation and that's going to benefit public health in the long term. In addition to that, during public health emergencies, there's going to be a discrete role for them working with public health. So they'll have in place they will understand. Ideally, these academic centers will be working with public health agencies, so they'll understand how things work. They'll have in place the data use agreements and material transfer agreements and so forth that they need to work together. This is one of the secrets that made the UK program work so successfully. They had programs like this in place, and then in addition to that, there's an educational component where we want them to take the lead on, for example, on training epidemiologists. So we and state and local public health agencies would work with them on the curriculum, but they would help translate this into online training for epidemiologists, both here in the U.S. and hopefully elsewhere in the world.
Yeah, that's important. That was the other topic I wanted to touch on, was the training, because we know we don't have enough people out there trained in epidemiology and genomics to conduct the work, analyze the data, and then help it make sense to all of us as we try to understand what's going on. So I appreciate those comments too. So thank you, Dr. Armstrong, so much for answering my questions. We have a little bit of time left, so I'm going to open it up to questions. If there's any questions from the audience, to ask a question, you can locate the Q&A button on the bottom of your screen and type a question there. So let's just go see if we have any questions.
And while we're waiting, it'll be announced at grants.gov, I believe it is, or grants.gov. The standard place universities look for granting, but we'll also make a lot of announcements when the competition does open up.
Okay, great. I don't see any questions in the Q&A, and we're at the top of the hour. So thank you very much, Greg, for joining us today. We really appreciate you being here and answering all of our questions. But seeing no more questions so far, I want to thank everyone for attending. This will conclude the program. Again, thanks for attending. And thank you, Dr. Armstrong.
Thanks to ASM and very much thanks to Congress for being so supportive.